Hi, folks. So, uh, first, I'd like to talk about further on the topic of formation of cancer. If you haven't already, make sure to like, share, subscribe, and click on the bell icon. Uh, when a cell, when a tumor tissue is detectable, it generally reached about 1 gram in its weight and about 10 raised to 9 cells, that means 1 with 9 zeros, that many cells have been formed, like that's like 30 doubling, approximately 30 times. One cell is, so one cell is formed into 2, 2 into 4, like doubling 30 times. And there are a few things about when these tumor cells are formed. It's a certain clone of a mutated gene, or rather a damaged gene, which is forming. And as that clone is, further subclones are forming, and more further mutation within those clones. And it's like a sort of a survival of the fittest within these tumor cells. The newer cells that are forming, many of them will be removed by the body itself, or they will just die, but the ones that are the fittest will actually remain. Uh, that is also referred to as tumor progression. Tumor progression is like when the fittest cells are remaining, and those fittest cells are also the most damaging cells, most malignant cells. They have the maximum potential to further proliferate, multiply. If that is, the if they are the type of cells which will be malignant, and that's when it's called tumor progression. Uh, and another thing about this is that when, like I mentioned, that the different mutated cells are changing, more and more different types of clones are forming. It's the When new clones are formed, it's like a sort of almost like a change in the species. In or in when different species are formed, it takes millions, possibly thousands or millions of years. But in tumors, the genes alteration are happening so fast within months, weeks, years, that it's almost like a different species is forming, of type of cells are forming, and but they are just going on and on proliferating, and within them the most damaging cells are surviving. The thing about this is that these fittest cells can, like, if you take one of, at this stage, whichever stage of the cancer may be, and you take out the most fit cell of all of them, and you view them by whether it be the DNA sequencing or any form, their gene, or you will be able to observe uh, all the way up till the initiating mutation, the driver mutation, or the first mutation that came, and that led to all these new sub. So it can be the subclones of the family tree can be seen all the way up till the starting point. And with this, you know, especially even, even after the, like, a treatment is formed, uh, if there is there is a certain cancer therapy is given, and those cells have mutated, now further mutated after the treatment, you can also still see that the tumor cells which are formed prior to treatment, their genetic alterations after the treatment can be observed. And that's the functions of that's how, uh, that's the way they behave, these type of cancer cells, and that's the way they are forming. So that's something I wanted to share as well. Thanks a lot. Cheers.